molecularly iodine is very, very small. Starch, on the other hand, when it's in a liquid form, it's clear and it's really, really big. Okay, so we have a small molecule, we have a much larger molecule. What's interesting about these two molecules is that when starch and iodine meet, they form this purple color. So we can call iodine an indicator of starch because it will turn purple when it's in the presence of starch. So here's what iodine looks like when it's in a test tube. And so you see this has kind of a yellowish reddish color. Starch, on the other hand, is this clear color when it's in liquid test tube. So if I take one drop, or maybe a couple of drops of the starch, or I'm sorry, the iodine, and put it into the starch, immediately, as soon as the starch and the iodine meet, you get this purplish color being formed. Okay. So I'm going to set up an experiment to test diffusion and test my knowledge of diffusion now. So I've got this beaker, and in this beaker I have just a little bit of water, okay? And I'm going to add a couple of drops of iodine to the outside of the beaker. So first I need to put on my safety gloves. I'm always following my safety rules. Okay, and here's my bottle of iodine. So I'm going to put just a couple of drops in here because iodine is really potent, and as soon as I put it into the water, I'm just not going to need very much. Okay. So now I have iodine and water mixing in this beaker right here. And let's just mix it up just a little bit. So water and iodine on the outside of the beaker. And now we have something that I hope that you've also used in biology. This is called a dialysis bag. And this bag has really, really thin pores in the bag. So essentially this is going to serve as a model for a cell, and we're going to pretend like the bag, the outside of the bag, is this semi-permeable membrane. So even though you can't really see the pores because they're so tiny, things that are small enough can move into and out of the bag. So I tied a knot in one end of the bag, and then I can open up my bag and I can put some liquid inside. So I'm actually going to go ahead and put starch inside this semi-permeable bag. So I'm going to put just a couple droppers full of starch into this bag. And as soon as I've got that set up, I want you to start thinking about what you predict will happen in this experiment. So while you're watching me do this, think about where you think the iodine could move and whether or not this iodine could move into the bag, and where you think the starch will move. Can the starch get out of the bag? And because you know starch and iodine form this purple color, do you think we'll see any color changes? Okay. So now I've got starch in my bag, so I can tie a quick knot in it. My gloves don't get in the way. And then I'm just going to rinse off this bag really quickly to make sure I didn't get any kind of iodine or any kind of contamination. If you look, I actually see a couple of drops of purple right now in my beaker, which tells me that I probably already slightly contaminated the iodine with a couple of drops of starch. But it should be alright, because I only see a little bit in there. So now I'm going to soak my dialysis bag in the iodine, and I'm going to let it sit for about 15 or 20 minutes. Can we pause for just a second? Alright, so we're back, and our experiment had a little bit of time to run. So before we look at the experiment, let's take a minute and answer our prediction questions. So the first one said, do you think iodine can permeate? In other words, move into the bag? And quite frankly, personally, I said, yep, I think it can, because I told you at the beginning, this is a really small molecule. So if iodine is small enough, it's capable of moving through the pores to get into the bag. Do you think starch can get out of the bag? Why or why not? Well, based on what I told you earlier, no, I don't think so, because I know that big molecules are going to get blocked from entering and leaving, so I think the starch is so big that it's going to be trapped. So in other words, now I think that iodine can move into the bag, but starch is going to stay trapped inside. So if that's the case, and iodine can move into the bag, what we should see is a color change And we should see the bag go from clear to purple because iodine and starch have met. On the other hand, if starch can't get out of the bag, there should be no color change in the liquid outside of the beaker. So take just a moment and think through that. Look at numbers 3 and 4 for predicting the color of the bag and the beaker, and just check to make sure that makes sense. And if you need to turn to a partner and make sure that you can explain it, that's absolutely fine, because it gets a little bit tricky when we're thinking about the movement of starch and iodine at the same time. Okay, so hopefully you've had a second to just predict the color of the bag, predict the color of the beaker, and now I can show you. So after our experiments, the starch inside the bag has turned this brilliant, beautiful purple color. And that's because the iodine was able to get into the bag, and you start to see this color change. Because remember, I told you iodine is an indicator of starch. On the other hand, there's absolutely no color change in the beaker at all. So the iodine that was outside of the beaker must not have ever met with starch, so starch must have stayed trapped inside the bag. So now you have a beautiful example of the membrane being semi-permeable because it's permeable to iodine, but it's not permeable to starch. You also see diffusion here because you know that I put a ton of iodine outside of the bag. So outside of the bag was my high concentration of iodine. Inside the bag was just starch, so that was my low concentration of iodine. So iodine was able to move into the bag and make this color change. So there you have it. In a nutshell, osmosis, diffusion, and cellular movement. Good luck on your HSAs.
Body systems, what you need to know. There are 8 major body systems tested on the HSA. The nervous system, the circulatory system, the respiratory system, the endocrine system, the digestive system, the excretory system, the skeletal system, and the muscular system. The nervous system consists of the brain and the nerves that connect the brain to the rest of your body. It controls movement, senses, hormones, circulation, respiration, and homeostasis. The circulatory system. This is your heart, veins, and arteries. It takes oxygen and food to the cells and moves carbon dioxide and waste products away from the cell. The respiratory system includes your nose, mouth, trachea, and lungs. This is where the exchange of carbon dioxide and oxygen occurs. It works in conjunction with the circulatory system. The circulatory system gets the oxygen from your lungs and takes it to your cells and returns the CO2 to your lungs from the cells after cellular respiration occurs. The endocrine system. This maintains your hormone balance in your body. It works with the brain to keep homeostasis in your body. And the hormones produced by the endocrine system are transported by the circulatory system. The digestive system. This includes the mouth, esophagus, stomach, and intestines. It breaks down the food into smaller particles that can be transported to the cells by the circulatory system. The excretory system. This is your intestines, kidneys, and bladder. It removes waste products from the body. The skeletal system. This is your bones. This is the skeletal system is what supports your body. It works in conjunction with the muscular and nervous systems to allow movement. The muscular system. The muscles are like the bicep, triceps, heart, and tongue. It works in conjunction with the skeletal and nervous systems to move the body. Last but not least, how do they all come together? All the body systems come together at the brain. The nervous system controls all other body systems. When you answer questions on the HSA about the body systems, remember that the brain controls all the other systems, and the brain is part of the nervous system. Good luck!